Okay, so no microemergence topic is complete without talking about enzymes. Enzymes are protein catalysts produced by cells to accelerate chemical reactions. This is basically what enzymes are right here. They are almost always proteins. At least one or two catalysts in cells are RNA. That's been shown on ribosomes. But all in all, most of them are proteins. Enzymes are what makes us alive. Enzymes are not structural, they're functional. So they don't give us shape, but they give us action. They catalyze things like glycolysis, the Krebs cycle, the ETS. When I say catalyze, I mean they make chemical reactions take place at an acceptable rate that otherwise would take place too slowly or not at all. Catalysts in general are chemicals that accelerate a reaction but do not directly get consumed by the reaction. So they are sort of like screwdrivers. Screwdrivers can put thousands of screws into wood to attach things together, but they do not become part of the wood or the machine or the apparatus that's being made with the wood. So a different enzyme is required for each chemical reaction that's going on in a cell. They're very specific. They can only do one specific thing because of shape requirements. Molecular activities in a cell all require different enzymes. And basically every cell and organism depends on thousands of special enzymes to make chemical reactions take place. One of the most important parts of an enzyme is its active site. In this picture that's right here, it looks like kind of the belly of a turtle. The active site's job is to actually bond to a substrate. So what you see here, this is a substrate. And when two of these actually stick in this active site here, you get a product. Or when this substrate, I should say, sits in this active site, it is turned into a product. This substrate fits into the enzyme with what we call an induced fit model, where the enzyme slightly changes shape and the substrate slightly changes shape as they meld or mesh with each other. And then as a result, some covalent bond is stressed inside the substrates, and then these two products are made as a result of the stressing of a covalent bond. Note that the enzyme here is not changed all the way through the process. It does change shape a little bit here, but it's not consumed by the process. The active site is really important. It's the part that actually binds to a substrate. A substrate is any chemical that actually binds to an enzyme, and the product is what's produced as a result of the enzyme interacting with the substrate. Let's see, cofactors are often needed by enzymes. A cofactor is like usually an ion that will sit like in here in the active site, and it allows the active site and the substrate to interact properly with the active site. So cofactors are not consumed. They're kind of like enzymes. They're not consumed by the reaction, but they're needed for the reaction to take place. So for example, in saliva, minerals, I can't remember exactly which, but maybe sodium is responsible for being a cofactor for amylase. Amylase converts starch to maltose. It's either magnesium or calcium. It's one of the basic minerals. Without that mineral in the mouth, even though the enzyme's there when you're eating a cracker, the starch in the cracker can't become maltose. So cofactors are kind of important for enzymes to function. The enzyme can then only be active when it has the substrate and the the cofactor needed to work. A coenzyme is different than a cofactor in that a cofactor is usually just a mineral, whereas a coenzyme is an organic molecule, like a vitamin. Vitamins are usually coenzymes. They're not enzymes, they don't catalyze reactions, but they work at active sites to help chemical reactions take place. They don't get consumed by the reaction, but they help the reaction go forward. The way an enzyme works is it actually lowers the activation energy. This is very important right here. The enzyme lowers the activation energy required for the reaction to occur. And what it comes down to is molecular speed is often the requirement for making reactions take place. And molecular speed in a typical reaction is usually has to be really high. What the enzyme is doing is it's stressing a covalent bond so that a rather moderate collision will actually cause the substrate to break into products. So normally only fast-moving molecules can participate in reactions, but what the enzyme does is it stresses bonds so that low-speed molecules will actually be able to participate in the reaction. Therefore, since more collisions in a system are moderate speed collisions and not high speed, the reaction actually goes much faster. And that's how all catalysts speed up reactions. They simply make it easier for the substrates to become products in terms of molecular collision speed. Now, here's a few animations of how enzymes work. In the upper left here, what we have is a substrate in pink binds to the active site of an enzyme. A stray molecule, just with a regular ordinary collision, hits the substrate, and there is sufficient stress in the middle with the covalent bonds that the moderate collision is capable of hydrolyzing the substrate into two products. This is a hydrolysis reaction here. And you can see that because it's consuming water. Water is not a cofactor here, and it's not a coenzyme. It's actually being consumed by the reaction. Notice the enzyme is not consumed by the reaction, but the substrate is turned into products, so it's consumed, and the water is consumed. The stray molecule is not consumed. Over here we have condensation. Two different substrates bind to the active site. A moderate collision hits them, and right out here where they touch, some covalent bonds are stressed. Water is kicked out as like fragmentary atoms from the collision, just like when two cars hit each other, pieces fall off. But then the two substrates are stuck together because they mesh a covalent bond. Okay. Now at the atomic level, if you really want to see right down to the atoms what's going on the left-hand side over here, this is hydrolysis. So this is hydrolysis. This was a polypeptide, and what the enzyme's doing is it's getting into the peptide bonds and it's cutting them apart. These water molecules are being consumed as that happens because hydrolysis requires water to seal up the bonds. This OH and this H come from the water. So when these are split apart, you see that OH there and that H there came from ultimately water molecules providing the atoms to seal up the severed covalent bonds. This is condensation. I'm sorry, this is hydrolysis, separating monomers from one another by using an enzyme and water. This is condensation. Condensation is putting monomers together to make polymers, and in this animation on the right, what we have is an enzyme is getting into the spaces between these molecules, and it stresses these H's and OH's here, knocks them off in a collision, and then an oxygen bridge forms between the two, and the two monomers are locked together. That is what condensation is. In all cases, it's basically an enzyme grabbing onto the H of one molecule and the OH of another, creating a stress so that the H and OH will come off as water, and the two molecules then form a covalent bond bridge between themselves. All right, so now we got to talk briefly about inhibitors. Turns out enzymes are vulnerable to interference by other molecules that are kind of doing the opposite of a catalyst. In this first case, what we have is a competitive inhibition. Here's the normal substrate. It binds to the enzyme, and then a collision hits the enzyme and the substrate, and it breaks into two products. These are products here. This molecule is not the same. This is like carbon monoxide. This is like oxygen. And oxygen normally is bound by an enzyme in the electron transport system, and it's used to make water. Carbon monoxide can also bind at that same spot on the active site, but when it does that, it will not become water, and instead it goes in here and it prevents oxygen from coming in. As long as this this inhibitor can actually release and sometimes allow the enzyme to come in, we call this competitive inhibition. So ultimately, the substrate and the inhibitor compete for the active site of the enzyme, but reactions only take place when the real substrate combines. Now that's different than this. Non-competitive inhibition is where a molecule either binds at the active site so strongly that it's permanently blocked, or worse yet, it binds to a different part of the enzyme and changes its shape. As a result, you'll notice the active site here has changed shape. As a result of the new shape of the active site, the substrate can't bind, the reaction can't take place. This is a disaster. So these two kinds of inhibition are basically seen in poisons. Almost all poisonings in the human body, whether it's snake venom or it's a plant chemical like cyanide, almost all of them are simply enzyme inhibitors. Some of them bind competitively, weakly or strongly. Others bind non-competitively and permanently disable the enzyme. So cyanide disables an enzyme called cytochrome oxidase in the mitochondrion. Snake venom, like cobra venom, disables and actually blocks a receptor. It's not truly an enzyme, but it blocks a receptor on neuron membranes, or in general just membranes, and prevents the transmission of nerve impulses to muscles. And that blockage, I believe, actually is at the neuromuscular junction. But still, it binds very strongly and prevents your chemicals from actually binding to where they're supposed to bind and cause a reaction. So most poisons are blocking substrates from doing their job and becoming products in the human body. Or I should say they block enzymes from doing their job. And that brings us to the last part here. There's a way to graph the activity of an enzyme. We call these path of reaction graphs. And on the left here, basically what we have is an endothermic reaction. And in an endothermic reaction, the reactants X have less energy than the product Y. And normally it takes a whole lot of energy to get X up to a really high energy state, and then it can convert to Y. This is called the activation energy. This entire hump from here to here is the activation energy. That's what this author is drawing right here. That's E sub A. So what an enzyme does is it basically, by binding to the substrate, it lowers E sub A so that the energy hill is not nearly as high. What is provided by the hump? The hump is basically collision speed. Really high collision speed is usually required to actually cause the enzyme, or cause the reactant to become the product. What the enzyme does is it binds to X. It makes it easier for it to become Y, so only a medium speed collision will convert it to Y. As it turns out, this graph is actually describing pH on the x-axis versus enzyme activity. It turns out every enzyme has an optimal pH level for its operation, and that's because changing the pH of a solution changes the shape of the enzyme in the solution, because the enzyme has functional groups on the amino acids that are interacting with water molecules by hydrogen bonding and things. And when you change the pH, you're adding or subtracting hydrogen ions and hydroxide ions, and that changes the chemical profile that's around the enzymes and then changes their shapes. So some enzymes work at really low pH. Many enzymes work around 7, and then these are stomach enzymes work here. Most of your cellular enzymes and blood enzymes work here. And some digestive tract enzymes work at alkaline pHs above 8. And then lastly, enzymes generally have an optimal temperature profile. In other words, for the human body, optimal temperature is around 37 Celsius. If you go much higher, the enzymes at the higher temperature actually start to break down, and that's called denaturing. When they denature, they do not work anymore. So the human body has a critical temperature of about 107 degrees Fahrenheit. If the brain heats up more than about 107, critical brain enzymes denature, and at that point the brain cells die because they cannot process specific activities. So you'll notice, though, as we get colder, our enzyme activity declines. This is activity level, which would be ability to process substrate and make product. So that's why as we get cold, it gets harder and harder to move. Below about 88 Fahrenheit, the heart can't even beat strongly enough to keep the brain at a conscious level of activity. So enzymes are a big deal in cells, and they have all kinds of characteristics that have been under study for years. Lastly, enzymes fight a most basic chemical phenomenon in the world called equilibrium. Most chemical reactions in living things are not the completion reactions that you're probably familiar with from chemistry. Completion reactions are where you add substrates together and they just all make products. In living systems, chemicals, we'll just call them like A, can be turned into B, but B can also be turned back into A, sometimes by the same enzyme. It just depends on how much of B is around and how much A is around. So living things, to make A become B, if that's what a cell wants to do, what it has to do is have high levels of A and get low levels of B. In other words, as soon as B is made, get rid of it. That keeps allowing A to become B. Cells cannot defeat equilibrium. All they can do is work around it. Equilibrium, if you remember from chemistry, which you may not have studied, equilibrium is the concept that chemicals do not usually, in living things or in many systems, they do not go to completion. Like completion reactions are things like methane, CH4. Oops, let's start over again. CH4 plus O2 yields CO2 plus H2O. That's a completion reaction. This is how we heat our houses in the wintertime, and this is how we heat water for our hot water heaters in most houses. It's completion, meaning CO2 and water is never going to go back and form methane and oxygen. Methane and oxygen will react together to form carbon dioxide and water, but it won't go backwards. It's only a forward reaction. On the other hand, in living systems, you get lots of reactions like you see here on the left. Carbon dioxide, when you breathe it out, let's talk about inside of your cells. In the cells of your body where carbon dioxide is made, some of it reacts with water to form bicarbonate. The bicarbonate goes through your capillaries. Some of it actually goes on to form hydrogen ions and bicarbonate ions. When this is in your body cells, when your blood reaches your lungs, the lungs start to exhale carbon dioxide. In the lungs, the reverse reaction takes place. The reason this is, in the body cells, carbon dioxide is high, so the equilibrium moves to the right and produces bicarbonate. But in the lungs, carbon dioxide is low, so bicarbonate and protons react together to form carbonic acid, which in turn disintegrates into carbon dioxide and water, and we exhale the carbon dioxide out of the body. So let me summarize that one more time. In the body cells, carbon dioxide is being made by the Krebs cycle in your mitochondria. Its levels are generally high. It reacts with water, it forms carbonic acid, which in turn reacts to form protons and bicarbonate ions. When the blood with protons and bicarbonate ions reaches your lungs, suddenly this drops because in the lungs carbon dioxide is being exhaled. That causes these two to react together to form carbonic acid, which in turn reacts to form water and carbon dioxide, which is then being exhaled in the lungs. This equilibrium seesaw goes on constantly in human lungs until a patient dies. When somebody dies, the equilibrium becomes static. In other words, the amount of CO2, the amount of water, carbonic acid, proton, and bicarbonate become stable. They're not equal, they just become stable. They're never stable in a living organism. In a living organism, this is always high in tissues, low in lungs. This is always low in lungs, high in tissues. So the equilibrium seesaws back and forth. All right, so that concludes the discussion of enzymes. It's now time to talk about organisms and organs.